Welcome to the deep dive. Today we're getting into something vital, something complex, and honestly, sometimes terrifying, your immune system. And we're going to start with a really stark example. The Ebola outbreak back in 2014. Remember that? West Africa, highly contagious, this hemorrhagic fever.
Yeah, devastating. Fatality rates of 50%, sometimes even higher. Thousands lost their lives.
Exactly, a truly relentless virus. So the big questions are, what makes something like Ebola so lethal? And maybe more importantly, how does your body even try to fight back?
Well, the mechanism is quite insidious, actually. Ebola usually spreads through direct contact, right? It gets into your system and targets specific cells, mainly your white blood cells.
Okay.
It binds to receptors on the cell surface, gets inside, and then, well, it basically co-ops the cell's own machinery.
Co-ops, like takes over.
Completely turns it into a little virus factory, churning out copies of itself. And eventually, that host cell is destroyed in the process.
What?
And because it targets those white blood cells, which travel through your lymphatic system, the virus can spread really, really quickly throughout the body.
And isn't there this thing, the cytokine storm? I remember hearing about that.
Yes. That's a critical and tragic part of the story. Late in the infection, as tissue damage gets severe, your immune cells can sometimes just unload a massive, uncontrolled release of pro-inflammatory signals, cytokines.
So your own immune system goes haywire.
Exactly. It's this overwhelming burst that, paradoxically, contributes hugely to the high fatality rate. Your defenses go into overdrive and end up causing more harm.
That's terrifying. But efforts are ongoing, right? Vaccines, treatments?
Oh, absolutely. There's constant work on vaccines, on antibody-based therapies, and just trying to understand how these pathogens manage to evade our defenses in the first place.
Which brings us to today's deep dive. We're exploring this incredible complexity, the immune system itself, how it fights invaders like Ebola, how it learns, and yes, how it can sometimes go wrong. We'll be drawing on some really foundational research here.
That's the plan. We're going to unpack how your body constantly performs this molecular screening, figuring out self versus non-self, and how it launches these incredibly specific attacks. But also, why this powerful system isn't foolproof.
Okay, let's start with the basics, the big picture. There are two main arms to the immune system, right? Innate and adaptive.
Correct. Innate immunity is your immediate sort of built-in non-specific defense. Think of it as the first responders on the scene. Always ready, acts fast.
And adaptive.
Adaptive is the more specialized force. It's acquired, it's highly specific to particular pathogens, and crucially, it remembers. That's why vaccines work. They train your adaptive immune system.
Got it. And these systems need different strategies depending on the enemy, like viruses inside cells versus bacteria outside.
Precisely. Intracellular pathogens, like viruses hiding inside your cells, need a different approach than extracellular ones, like most bacteria floating around.
And the weapons, you mentioned cells and proteins.
Yep. Specialized cells that can engulf or kill infected cells, and soluble proteins like antibodies that can neutralize pathogens or tag them for destruction.
And this all happens in specific locations, lymphoid organs.
Right. Places like your bone marrow and thymus, those are the primary lymphoid organs where immune cells develop. And then secondary sites like the spleen, lymph nodes, even tonsils where immune responses are often initiated.
Okay, let's focus on that first line, innate immunity, the immediate response.
So when a pathogen gets past your physical barriers, like skin, it often first encounters phagocytic cells. Think macrophages, dendritic cells. They're like the security guards, patrolling the tissues.
How do they know what's bad?
That's where a truly groundbreaking idea comes in, from Charles Janeway back in 1989. He proposed these things called pattern recognition receptors, or PRRs.
Pattern recognition, okay.
Yeah, these are sensors on immune cells that recognize molecules absolutely essential for microbes, but totally absent from our cells. Think like components of bacterial cell walls or viral genetic material. It's a way to spot generic danger signals.
And the most famous of these are the toll-like receptors, the TLRs.
Exactly. And the story of their discovery is fascinating. It actually started with fruit flies.
Fruit flies? Seriously?
Seriously. In the mid-90s, Jules Hoffman's group in France found these mutant flies. They were incredibly susceptible to fungal infections. Turned out they lacked a protein called toll.
Which they thought was just for development, right?
Right. But Hoffman linked it to immunity. Then, Janeway and Ruslan Medzitov found the human equivalent, TLR4. And Bruce Butler linked TLR4 specifically to recognizing LPS, a major component of many bacteria.
Wow. So a flight gene leads to understanding human bacterial detection.
It's a beautiful example of conserved pathways. We have at least 10 functional TLRs in humans. Some sit on the cell surface, watching for extracellular microbes. Others are inside, in compartments called endosomes, scanning for things the cell has ingested.
And they recognize all sorts of microbial bits.
All sorts. Bacterial cell wall components, flagellum from bacterial tails, viral double-stranded RNA, bacterial DNA, a whole range. When a TLR gets triggered, it sets off signaling cascades inside the cell, basically yelling, "Invader," and initiating an immune response.
And this has real-world impact. You mentioned drugs.
Yeah, some drugs, like Eldora, used for certain skin conditions caused by viruses, were actually found later to work by stimulating specific TLRs. They essentially kickstart the local innate immune response.
Okay, that's innate immunity. Quick, non-specific. Now let's shift to adaptive immunity, the specific learning part. Only in vertebrates, you said.
That's right. It offers specific, long-lasting protection. And we usually divide it into two main types:
Which are?
humoral immunity and cell-mediated immunity. Humoral is mainly about antibodies produced by B lymphocytes or B cells. These antibodies are great against extracellular threats, things floating in your blood or tissues, like toxins or viruses before they get into cells.
And cell-mediated.
That's the domain of T lymphocytes, or T cells. They don't make antibodies. Instead, they recognize and kill your own cells that have become infected or cancerous. They're crucial for dealing with those intracellular pathogens.
B cells for outside, T cells for inside. Simple enough. And they both start in the bone marrow.
Both originate from the same stem cells in the bone marrow, yes, but they mature in different places. B cells mature right there in the bone marrow. T cells travel to the thymus, the gland behind your breastbone, for their maturation.
Ah, okay, so they're trained separately.
Exactly. And this separation explains certain immune deficiencies, where someone might lack B cell function but have normal T cells, or vice versa.
So how does this adaptive system learn? You mentioned specificity. That involves antigens, right?
Right. An antigen is basically any foreign substance, usually a protein or a polysaccharide, that can trigger an adaptive immune response.
How does the body know how to make the right antibody or activate the right T cell for a new antigen it's never seen before?
That was a huge question. For a long time, the thinking was sort of instructive, like the antigen somehow shaped the antibody. But Niels Jerne, in 1955, proposed a radical selective idea.
Selective.
He suggested your body doesn't learn to make antibodies on the fly. Instead, it already has a massive pre-existing library of B cells, each making a slightly different antibody. When an antigen comes along, it simply selects the B cell with the antibody that happens to fit it best.
Like finding the right key for a lock that already exists.
Precisely. And S. McFarlane Burnett expanded this into the full clonal selection theory just a couple of years later, and it became the central pillar of immunology.
Okay, so break that down. What are the key points of clonal selection for, say, B cells?
Sure. First, commitment before antigen. Each B cell, through some amazing DNA rearrangements we can talk about later, commits to making only one specific type of antibody. This happens before it ever encounters its target antigen. You end up with millions of B cells, each with a unique receptor.
So you have this huge repertoire ready to go.
Exactly. Second, antigen selection. When an antigen finally appears, it binds only to the B cells displaying the antibody that fits it. That specific binding is the selection signal.
The key finds its lock.
Right. Third, clonal expansion. The selected B cell gets activated and starts dividing like crazy. It makes thousands, millions of identical copies of clone.
An army of specialists.
Yep. Fourth, differentiation. Most cells in that clone become plasma cells. These are basically antibody factories, churning out huge amounts of the soluble form of that specific antibody into your bloodstream.
And some become memory cells.
That's the fifth point. A fraction of the clone becomes long-lived memory B cells. They stick around, sometimes for decades, ready to mount a much faster and stronger response if the same antigen shows up again. This is immunological memory.
Like those 1918 flu survivors still having cells decades later.
Incredible, isn't it? And the sixth, absolutely crucial point is immunological tolerance.
Preventing attacks on yourself.
Exactly. During B cell development, any cells whose antibodies happen to react strongly with your own body's molecules' self-antigens are normally eliminated or inactivated. This prevents autoimmunity.
Sometimes that tolerance breaks down, right? That leads to autoimmune diseases.
Unfortunately, yes. Despite the safeguards, sometimes self-reactive lymphocytes slip through, or tolerance mechanisms fail later in life. When that happens, your immune system mistakenly targets your own tissues.
And that causes diseases like MS, type 1 diabetes.
Multiple sclerosis, where the myelin sheath around nerves is attacked. Type 1 diabetes, where T cells destroy insulin-producing beta cells in the pancreas. Graves' disease, hyperthyroidism from antibodies stimulating the thyroid, rheumatoid arthritis attacking the joints, lupus, or SLE, a systemic disease where antibodies target components inside cell nuclei.
Wow, a whole range of targets.
It really is. Also things like inflammatory bowel diseases, Crohn's, ulcerative colitis, where the immune system seems to overreact to normal gut bacteria.
Is it genetic or environmental?
It's complex. There's definitely a strong genetic link, often involving specific versions of those MHC genes we mentioned earlier, but it's not purely genetic. Studies on identical twins show that if one twin has an autoimmune disease, the other doesn't always get it. So, environmental triggers, infections, maybe epigenetic factors play a role too.
But we are getting better at treating these.
Hugely better. We've moved from broad immunosuppressants, which just dampen the whole immune system.
Increasing infection risk, I imagine.
Right, to much more targeted therapies, trying to restore tolerance specifically to the self-antigen involved, or blocking specific inflammatory molecules like TNF-alpha and rheumatoid arthritis drugs like Remicade or Humira do that.
What about targeting the immune cells themselves?
But that's another major strategy, using monoclonal antibodies to deplete specific cell types, like B cells. Drugs like Rituxan, originally for lymphoma, turned out to be surprisingly effective in some autoimmune diseases like RA and MS.
Interesting that getting rid of B cells doesn't always cripple your ability to fight infections.
It is surprising, but often T-cell immunity and existing antibody levels can compensate adequately. There are also drugs that block pro-inflammatory cytokines, or even treat with other cytokines, like using interferon beta for MS. And some newer drugs block immune cells from migrating to sites of inflammation.
Like preventing the attackers from reaching the target.
Exactly. Drugs like Tysabri for MS work that way, though they can carry risks. And for very severe cases, hematopoietic stem cell transplantation can sometimes reset the immune system, but that's a very high-risk procedure.
Okay, shifting gears again, let's talk about a huge success story. Vaccination.
Ah, yes. Edward Jenner, 1796. The classic story.
The milkmaids and cowpox.
That's the one. Jenner observed that milkmaids who got cowpox, a mild disease, didn't seem to get the deadly smallpox. So he did his famous and ethically dubious by modern standards experiment.
Inoculating the boy.
He took pus from a cowpox sore and inoculated an 8-year-old boy, James Phipps. Later, he deliberately exposed the boy to smallpox, and Phipps didn't get sick. Jenner proved that exposure to the related, milder virus conferred immunity.
And vaccination comes from vacca, Latin for cow.
Precisely. The principle is inducing active immunity. Take the tetanus vaccine, for example. You're injected with tetanus toxoid. The harmful toxin chemically modified so it's harmless but still recognizable by your immune system.
So your B cells see it.
Your B cells see it, get selected, proliferate, and make antibodies and memory cells against the toxoid. Those same antibodies will neutralize the actual toxin if you encounter tetanus bacteria later. That's active immunization. Your body actively builds memory.
And passive immunization is different.
Passive is just giving someone pre-made antibodies. Like if you get a nasty cut and might be exposed to tetanus but aren't up to date on your vaccine, you might get an injection of anti-tetanus antibodies. It provides immediate protection, but it's temporary because your body didn't make them and there's no memory.
Okay, so we know that these things work, but how did scientists figure out the molecular details, like how T cells actually see the antigen? You mentioned MHC before.
Right, the major histocompatibility complex. It was initially discovered because of tissue rejection in transplants. Skin grafts between unrelated individuals fail because their MHC molecules are different, recognized as foreign.
It's like your body's unique ID card.
A very polymorphic one. The human MHC locus, called HLA, is one of the most variable parts of our genome. Thousands of different versions, or alleles. That's why finding matching organ donors is hard, and why transplant recipients need immunosuppressants like cyclosporin A.
But its normal job isn't rejecting transplants, right?
No, its crucial normal job is antigen presentation. It's how T cells see what's going on inside other cells.
How is that figured out?
Through some really elegant experiments. Zinker, Nagel, and Doherty, back in 1974, were working with mice infected with a virus called LCMV. They found that cytotoxic T cells, the killer key cells from an infected mouse, could only kill other infected cells if those target cells came from a mouse with the same MHC type.
So the T cell had to recognize both the virus and the right MHC.
Exactly. That was the discovery of MHC restriction. The T cell sees the foreign antigen in the context of self-MHC. Other experiments showed antigens needed to be processed first.
Processed?
Yeah, broken down into smaller peptide fragments inside the cell, usually by cellular machinery in compartments like lysosomes. You couldn't just stick the whole antigen on the outside of the cell. It had to be internalized and chopped up first.
And then these fragments bind to MHC.
Precisely. The evidence pointed strongly towards a physical association. And then came the structural breakthroughs, mainly from Don Wiley's lab and others, using X-ray crystallography in the late 80s and early 90s. They solved the 3D structure of MHC molecules.
What did they look like?
MHC class I molecules, the ones that display peptides from inside the cell, have this amazing groove on top, formed by parts called the alpha-1 and alpha-2 domains. It's about 25 angstroms long, 10 wide.
And the peptide fits in there.
It sits right down in the groove, in an extended shape. Crucially, the peptide mainly interacts with conserved parts of the MHC groove's backbone, meaning one MHC molecule can actually bind and present many different peptides, as long as they have the right general shape and maybe a few key anchor amino acids.
So it's versatile.
Very. And the T-cell receptor, the TCR on the T-cell, then comes along and recognizes the combination, both the specific foreign peptide nestled in the groove and parts of the self-MHC molecule holding it. It's a ternary complex.
Okay, so T cells, like B cells, are clonally selected too, each with a unique receptor.
Yes, exactly the same principle. Each T cell has a unique TCR generated by similar gene rearrangements as antibodies, and they get activated when their TCR recognizes its specific peptide MHC complex on another cell.
And those presenting cells are called APCs.
Antigen-presenting cells. While most cells can present antigens via MHC class I if infected, there are professional APCs, like dendritic cells, macrophages, and B cells, which are especially good at activating T cells, particularly helper T cells.
Dendritic cells, you said they're the sentinels.
Yeah, they hang out in tissues like your skin and linings. When they encounter a pathogen, they gobble it up, process its antigens, and then they mature and travel to the nearest lymph node.
To report for duty.
Kind of. In the lymph node, they present the antigen fragments on their MHC molecules to cruising P cells. If a T cell with a matching TCR comes along, it gets activated.
And that leads to clonal expansion again. That's why lymph nodes swell.
That's exactly why. The activated T cell starts dividing rapidly. You get this massive proliferation. After the infection is cleared, most of these activated T cells die off apoptosis, but some remain as long-lived memory T cells.
How do T cells actually do their jobs?
Two main ways: direct cell-to-cell contact and releasing those chemical messengers, cytokines. Cytokines are incredibly potent. They can tell other cells to divide, differentiate, secrete other cytokines. It's a complex communication network. There are also chemokines, which mainly act as guides, telling cells where to go, like directing them to sites of inflammation.
And you mentioned two main types of T cells earlier.
Broadly, yes. Cytotoxic T lymphocytes, or CTLs, and helper T lymphocytes, TH cells.
CTLs are the killers.
They're the assassins, yes. They typically express a protein called CD8 on their surface, so they're often called CD8 plus T cells. Their job is to screen your body's cells, looking for signs of trouble presented on MHC class, like viral infection or cancerous changes. If they see something wrong, they kill that cell directly, usually by inducing apoptosis, programmed cell death.
How do they trigger apoptosis?
Couple of ways. They can release proteins called perforins, which punch holes in the target cell membrane, and granzymes, enzymes that enter through those holes and trigger the death cascade. Or they can directly bind to death receptors on the target cell surface. They're crucial for clearing intracellular infections.
And helper P cells, T8 cells.
These are the orchestrators, the activators. They usually express CD4, so they're CD4 plus T cells. They don't typically kill cells themselves. Instead, they get activated by professional APCs presenting antigen on MHC class II molecules. Once activated, TH cells release cytokines that help activate other immune cells, including B cells to make antibodies, and CTLs to become more effective killers. They coordinate the whole adaptive response.
And that's why HIV is so devastating. It targets these helper T cells.
Precisely. HIV infects and destroys CD4 plus T cells. As their numbers plummet, the entire adaptive immune system loses its coordination, leaving the person vulnerable to all sorts of infections and cancers. That's AIDS. And even within helper T cells, there are subtypes Th1, Th2, Th17, each producing different sets of cytokines and specializing in helping different types of immune responses, like fighting intracellular bacteria versus parasites.
Okay, let's loop back quickly to antibodies. You said they're on B cells, but also secreted.
Right. On the B cell surface, they act as the B cell receptor, BCR, the antigen sensor. When secreted by plasma cells, they're the soluble weapons.
What do they do when secreted?
They can neutralize pathogens directly, like blocking a virus from entering a cell, or they can coat a bacterium, tagging it. So phagocytic cells like macrophages recognize and eat it. That's called opsonization. They can also activate another system of blood proteins called complement, which can directly kill bacteria or enhance inflammation.
And they have that Y-shaped structure.
Yep. Typically made of 4 protein chains, 2 identical larger heavy chains, and 2 identical smaller light chains, linked by disulfide bonds.
And different classes exist. IgG, IgM.
5 major classes in mammals: IgA, IgD, IgE, IgG, and IgM. They have different heavy chain constant regions, which gives them different structures and functions.
Can you give a quick rundown?
Sure, IgM is usually the first antibody made in an initial response. It's often a pentamer, 5 Y units joined together, very good at activating complement. IgG is the main antibody in your blood, dominant in secondary responses, lasts a long time, and crucially, it can cross the placenta, giving passive immunity to a fetus.
Ah, but that can cause problems too, right? Like with Rh factor.
Exactly. If an Rh-negative mother carries an Rh-positive fetus, her IgG antibodies against Rh factor can cross and attack the baby's red blood cells. IgE is involved in fighting parasites and also mediates allergic reactions. IgA is specialized for protecting mucosal surfaces, gut, respiratory tract. IgD, its function, is still a bit mysterious, mostly found on the surface of mature B cells.
And the specificity comes from the tips of the Y?
Yes, the variable regions, or V regions, at the tips of the heavy and light chains. Within these V regions are even more variable loops called hypervariable regions, or CDRs, complementarity determining regions. These form the actual antigen binding site. The rest of the chains, the constant or C regions, determine the antibody's class and effector function.
Okay, now for the really mind-bending part. How do we make so many different antibodies? Millions, potentially billions of specificities from a limited number of genes.
It's one of the most incredible feats of molecular biology. It stumped people for ages. The key idea came from Dreyer and Bennett in 1965, the DNA rearrangement hypothesis. They proposed that the V region and C region of an antibody chain were actually encoded by separate gene segments that were joined together at the DNA level during the B-cell development.
So the gene itself is assembled.
Exactly. And Susumu Konagawa provided the definitive proof in 1976. He compared the antibody genes in embryonic cells, where they hadn't rearranged yet, with those in mature antibody-producing cells. He showed that the V and C gene segments were far apart in embryonic DNA, but close together in the mature B-cell DNA. It was no prize-winning work.
So how does the rearrangement create diversity?
It's multi-layered. First, your germline DNA. The DNA you inherit doesn't contain whole V genes. It contains clusters of gene segments, multiple different V segments, several D diversity segments for heavy chains only, and several J joining segments.
Like building block.
Exactly. During B-cell development, a special enzyme system called VDJ recombinase randomly picks one V, one D for heavy chain, and one J segment, and joins them together, cutting out the DNA in between.
So each B cell makes a random combination.
A unique random combination for its heavy chain and another for its light chain. Just the sheer number of possible V, D, and J combinations generates enormous diversity, maybe millions of potential antibodies right off the bat.
But there's more.
Oh, yes. Second, the joining process itself is imprecise. The exact spot where the segments are stitched together can vary by a few nucleotides, creating slight variations right in the hypervariable regions. That's junctional diversity.
Little tweaks at the seams.
Right. And third, an enzyme called terminal deoxynucleotidal transferase, TDT, can randomly add extra nucleotides, N-nucleotides, at the junctions, particularly in heavy chains, further scrambling the sequence.
Wow. So combinatorial joining, imprecise joining, and random nucleotide additions.
It adds up incredibly quickly. Estimates suggest maybe 10, 11, or even more possible antibody specificities can be generated through these mechanisms before the B cell ever sees an antigen. T cells use a very similar VDJ recombination process to generate their diverse T cell receptors.
But wait, wasn't there something after antigen exposure too? Somatic hypermutation.
Ah, yes. This is crucial for refining the antibody response. After a B cell gets activated by an antigen and starts proliferating in structures called germinal centers and lymph nodes, another enzyme called activation-induced cytosine deaminase, or AID, gets turned on.
What does AID do?
AID introduces point mutations, single nucleotide changes, specifically into the rearranged V regions of the antibody genes. It basically peppers the antigen binding site genes with random mutations.
Why would it do that? Seems risky.
It is random, but it's coupled with selection. Some mutations will make the antibody bind the antigen less well, and those B cells will likely die off. But some mutations, by chance, will make the antibody bind better, higher affinity. Those D cells get stronger survival signals and are preferentially selected to expand and become plasma cells and memory cells.
So it's like directed evolution happening in your lymph nodes.
That's a great way to put it. It's how the immune response matures over time, leading to higher affinity antibodies and secondary responses. It explains why boosters often give you better, longer-lasting protection.
And class switching, changing from IgM to IgG.
Also happens in activated B cells, often directed by signals, cytokines, from helper T cells. The B cell essentially swaps out the constant region gene segment it's using, say, swapping the C-mu segment for IgM for a Phi gamma segment for IgG, without changing the rearranged VDJ segment.
So it keeps the same antigen specificity but changes the antibody's function?
Exactly. You can switch to IgG for better bloodborne activity, or IgA for mucosal sites, or IgE for allergies parasites, all while still targeting the same original antigen.
Okay, last major piece. How do these lymphocytes, B and T cells, actually get the go signal activation?
It's all about signals at the cell surface. Both the B cell receptor and the T cell receptor aren't just floating alone, they're part of larger protein complexes on the cell membrane. When antigen binds, it causes changes in these complexes that transmit signals into the cell.
And for T cells, you mentioned it takes two signals.
Yes, this is a critical concept for T cell activation, especially helper T cells. Signal 1 is the TCR binding its specific peptide MHC complex on the APC. That provides the specificity.
Know what to attack signal.
Right. But that's usually not enough on its own. It needs signal 2, which is called co-stimulation. This comes from interaction between other pairs of molecules on the T cell and the APC. The classic example is CD28 on the T cell binding to B7 molecules, CD80, CD86 on the APC.
Well, that can
It's a safety check. Professional APCs, like dendritic cells that have encountered pathogens, turn up their B7 expression. So the B7 signal tells the T cell, yes, the antigen you're seeing is genuinely associated with danger or infection. It's not just some harmless self-peptide.
What happens if a T cell gets signal 1 without signal 2?
It usually becomes unresponsive, a state called anergy, or it might even undergo apoptosis. This is a key mechanism for peripheral tolerance, preventing T cells from attacking healthy tissues that might occasionally display self-peptides but don't provide co-stimulation. Once a T cell receives both signals, it triggers a cascade of intracellular signaling events, leading to proliferation, differentiation, and cytokine production. One key cytokine T cells make early on is interleukin-2, IL-2, which acts back on the T cell itself, driving its own proliferation and autocrine loop.
Is it all just go signals, or are there breaks too?
Definitely breaks too. For example, another receptor called CTLA4 appears on T cells after activation. CTLA4 also binds to B7, but instead of activating, it sends an inhibitory signal. It helps to dampen the T cell response and maintain tolerance. Mice lacking CTLA4 die from massive T cell overproliferation.
A delicate balance. And how do helper T cells activate B cells?
It also typically requires linked recognition and multiple signals. The B cell first binds the antigen via its BCR, internalizes it, processes it, and presents peptides on its MHC class II molecules.
Acting as an APC itself.
Exactly. A helper T cell that recognizes that same peptide MHC complex on the B cell can then provide help. This involves direct contact, like the CD40 molecule on the B cell, binding to CD40 ligand on the T cell, and cytokines released by the T cell right onto the B cell, like IL-4, which can trigger proliferation and class switching.
It sounds like a very intimate conversation between the cells.
It really is, often happening in a specialized junction called the immunological synapse. Inside the cells, these receptor signals trigger complex pathways involving protein kinases, phosphatases, adapter proteins, all leading to changes in gene expression that drive the immune response.
Any particular signaling pathways worth mentioning?
Well, many pathways involved in lymphocyte activation rely on recruiting cytoplasmic tyrosine kinases, like Sandrasi family kinases, right after receptor engagement. For cytokine signaling, the JAK-STAT pathway was a major discovery.
JSTAT.
Yeah, Janus kinases, JAK, are associated with cytokine receptors. When cytokine binds, JAKs activate and phosphorylate transcription factors called STATs, signal transducers and activators of transcription. The phosphorylated STATs then move into the nucleus and turn specific genes on or off, mediating the cytokine's effects. It's a relatively direct pathway from receptor to gene expression.
Okay, wow. We've covered a huge amount of ground. From Ebola's attack to the body's innate alarm bells, the learning adaptive system, clonal selection, autoimmunity, vaccines, MHC, T-cell killers and helpers, antibody classes, genetic shuffling. It's incredibly intricate.
It really is. The sheer elegance of how your body distinguishes self from non-self, generates this unbelievable diversity of receptors, coordinates attacks, builds memory, and usually maintains tolerance. It's a molecular and cellular masterpiece.
And understanding it is key to tackling so many diseases.
Absolutely. Whether it's fighting infections, developing better vaccines, managing autoimmune diseases, or even harnessing the immune system to fight cancer, it all comes back to these fundamental principles we've discussed.
So as you go about your day, maybe take a moment to think about this constant microscopic battle happening inside you, this incredibly sophisticated defense system built through millions of years of evolution working to keep you safe. What aspect of it sparks your curiosity the most? Thank you for joining us for this deep dive into the immune system. Until next time, keep learning, keep questioning, and stay curious.